Hello everyone, welcome to the students presentation week 2025. Myself Manasvi Kamdi, student of B.Pharm 5th Sem of P.R. Patil Institute of Pharmacy, Talegaon, District Wardha. It starts the presentation on pharmacognostic study of asafoetida. The contents of this presentation are introduction, synonyms, biological source, geographical source, cultivation and collection, microscopical characteristics, macroscopical characteristics, solubility, chemical constituents, extraction and isolation, chemical taste, adulterants and substituents, therapeutic uses, and marketed preparation. The introduction are the plant of asafoetida is an herbaceous and perennial. Asafoetida is sulfurous-smelling gum resin that's extracted from ferula plants. It grows about 2 meters high with a circular mass of 30 to 40 centimeter leaves. These are the image of asafoetida plant. Then we see the synonyms. The synonyms of asafoetida are gum asafoetida, devil's dung, and hing in Hindi. Then we see biological source and geographical source of asafoetida. The biological source are the asafoetida is the dried latex extruded from the living underground rhizome or taproot of Ferula foetida, which is belonging to the family Umbelliferae. Then the geographical source of asafoetida is the mainly asafoetida plant are found in Asia, Afghanistan, and Kashmir in India. Then we see cultivation and collection of asafoetida. The asafoetida plant propagated by seeds require low rainfall and arid climate. Gum resin is collected from cuts made in roots and rhizomes. A milky juice exuded from the cuts. The juice is allowed to dry and it solidifies into a gum resin. The resin is then scraped off, dried, and packed in container. Then microscopical characteristics of asafoetida. The transverse section of oleogum resin contains resin canal scattered throughout the tissue. Resin canals, often filled with resinous material, appearing brownish or yellowish under the microscope. Fibers. Lignified fibers may be present, offering structural support. Starch grains, present in some fragments, simple, rounded or oval in shape, and may appear refracting. Oil globules, seen as small droplet within parenchymal resin canals. Calcium oxalate crystals may be observed as rapids or clusters, appear as slender needle-like crystals. Let's we see macroscopical characteristics of asafoetida. Color yellowish white changing to reddish brown. Shape. The asafoetida present in two different shapes, tears and masses. Tears are rounded. Size. The tears are 0.5 to 3 cm in diameter. Taste. Bitter and acrid. Odor. Intense and persistent. Then we see solubility. The asafoetida present in two types, red hing and hing carboxylate. Red hing is soluble in oil and hing carboxylate is soluble in water. Then chemical constituents. The chemical constituents present in asafoetida are the 40 to 64% resin, 25% endogenous gum, 10 to 70% volatile oil, 1.5 to 10% ash, sulfuric acid, umbelliferone, and many other constituents are present in asafoetida. Then extraction. The many steps involved in extraction of asafoetida. Firstly, collection of raw materials. The roots and rhizomes of Ferula asafoetida are cut to allow exudation of the gum resin. Then pulverization. The crude material is cleaned, dried and powdered to facilitate extraction. Then solvent extraction. Usually alcohol or ether is used to extract the resinous part. Then filtration. The solution is filtered to separate the insoluble part from the resinous extract. Evaporation. The solvent is evaporated under reduced pressure or at controlled temperatures to obtain concentrated resin extract. Last, purification. The extract may be further purified by repeated dissolution and precipitation method to enhance purity. Then isolation. Firstly, the selection of plant. The plant Ferula asafoetida is selected at maturity. Incision. Cuts are made in the roots or rhizome during the season. Collection of exudate. The resin is collected by hand as it pours out. It is sticky and pale yellow to brown in color. Drying. The resin is spread out, allowed to dry in the shade. This prevents loss of volatile oils and maintain quality. Then purification. Sometimes ethanol or water is used to extract and purify active constituent. Last is storage. The purified resin is stored in airtight containers to protect from moisture and contamination. Chemical tests. The chemical tests are performed to identify asafoetida drugs are the fractured surface of the crude drug, if treated with sulfuric acid, forms reddish brown color. When treated with 50% nitric acid, then they give green color. The asafoetida drug is added in the water, then triturate it gives yellow-orange emulsion. Then we see adulterants and substituents. Asafoetida is adulterated with gum arabic, rosin, red clay and barley. If any color compounds are mixed in pure asafoetida, then they are detected by simple soluble shake. Small amount of sample dissolve in sufficient amount of water in a test tube and shake. Pure asafoetida dissolve very quickly with milky white color, whereas colored compound forms colored solution. Then we see therapeutic uses. The asafoetida commonly has many therapeutic applications. It is used as a carminative, nerve stimulant, expectorant, antimicrobial agent and antibiotic activity. Because of its strong antibacterial and antiseptic properties, it is considered a natural antibiotic. The marketed preparation, the Hingavasta Churna by Baidyanath Ayurved Bhavan, is used to improve digestion, relieve gas, bloating and abdominal discomfort. Next is acknowledgement. I express my heartfelt gratitude to Dr. Nilesh Banarse sir, our esteemed guide, for his continuous support, motivation and valuable guidance. I am thankful to respected Principal Sir, Director Sir, non-teaching staff for their encouragement and assistance. Special thanks to my friends and classmates for their cooperation and moral support. Thank you. Have a nice day.